Hello!
Hi there.
Welcome to Microbiology Journal Club, where you nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker of pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aim the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week we meet to talk about microbiology, and today it's our news week where we're doing an overview of some of the coolest microbiology papers that we've seen during the week. Oh, well,
Yes,
now it's maybe been the month now, so it's like we're coming back after a bit of a hiatus.
yes, indeed. We both had various illnesses. Usually, we also at the end of the month we usually select a paper to do a deep dive in. So make sure to message us at MicroTWC if you want us to look at something that's caught your fancy.
Absolutely. You can follow along with any of the papers that we discuss in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you. So please use the comments or tweet us at MicroTWJC. And we are fully back after about a month. And boy, has stuff has happened while we were away.
So much stuff. The world doesn't stop when you get sick.
Exactly. We have a brand new outbreak from the disease called monkeypox, which we're going to look into about what this outbreak is going, what we know about this outbreak so far, and how it's been going on for maybe a lot longer than anyone thinks. But that isn't all. We've also got a swathe of new Omicron variants out there now and terrorizing lots of people, including at least one of my co-hosts.
Yes, yes. I'm coming back from.
But wait, there's more. We've also got some fantastic new studies visualizing how the microbiome lives in the gut, and we learn how some microbes swim using tinsel-like appendages.
Absolutely.
So
Yeah, so let's dive into this monkeypox story first. This is definitely something that I had seen in the news, and I'm so happy to have like Faz and you guys, the community, I guess, those who are watching, to come back and read some of these things because it kind of just like briefly touched my radar, and now we get to dig into some of the papers.
yeah, so monkeypox. It's a brand. Well, we're saying it's a brand new disease, but actually it was discovered in discovered in monkeys in 1958,
Yeah.
and the first case in humans was diagnosed in 1970 in a baby in like the Democratic Republic of Congo. Um,
Yeah,
and
it's a DNA-based virus. Um,
yeah,
so like that's in the same line as, well, smallpox is actually it's closely related to smallpox. It comes from the same family, orthopox viruses.
yes, and it's characterized by these like kind of pustules that appear on the skin, the
Mhm.
pox kind of rashes.
Yeah.
Uh,
Yeah, like
and
all
like
the things with pox, I think, have to do with the skin. Like chicken pox is like the same type of lesion, but the severity, the density of those skin lesions varies with the different types of poxes.
yeah, with this one, it starts off with like a fever and kind of like general illness symptoms, and the
Mm.
actual pustules start appearing maybe like 2 or 3 weeks into it.
Hmm.
And a person is considered infectious from the onset of illness until those lesions have like crusted over, or those crusts have like separated off, and a
Mm-hmm.
fresh human layer of like healthy skin is formed underneath the crust. So
Yeah.
human transmission is thought to only occur through direct contact with infected body fluids or lesions, or via like infectious kind of fomites and surfaces, through
Hmm.
respiratory secretions occasionally, and typically require prolonged like kind of contact in order to for it to happen. So it's not
Hmm.
like the most infectious of diseases. You have to be near to
Right.
someone for quite a while in order to have the opportunities. Um,
Right. Yeah. Like that's from that description, it sounds like it's not really like you like brush against something. It's not like from that action. That's like a smaller risk than maybe like hugging or something, or like maybe sitting beside people. I guess sexual contact, right, would be that's like extended skin contact sort of moments.
yeah,
Massages, right?
I
Like
mean,
that actually would be a route. Yeah.
the kind of impression I get is you get lots of viruses kind of just bouncing off of people and not getting infected. And there's only a small percentage of chance that it will actually cause an
Right.
infection. So the longer you're next to someone who's infected, the higher that probability gets.
Yeah. I do. I just want to maybe want to talk about like that. Just reminds me of how much I love the skin in terms of a defensive organ, right? Like it
Oh,
does a really
skin.
good skin. Yeah. uh, stuff bounces off all the time from us. But yeah, like it's not It's not a force field, I guess. It's as much of a force field as we get with biology.
Yeah, exactly. I mean, even in Star Trek, the force shields come down occasionally.
That's true. That's true.
So that brings us to 2022, where an isolated case was imported from a person who visited Nigeria, which is described in this study as an endemic country for monkeypox, which we'll talk about a bit more later.
Hmm.
And the first like laboratory confirmed case was on like the 7th of May, and it was very unusual, but not unheard of for monkeypox to have appeared in the UK. And so
Right.
they did some contact tracing to see what this who had been near to this person, and they identified 116 different contacts, including healthcare workers, but none of those contacts developed monkeypox infection during the 21-day follow-up period.
Mm.
However, that people weren't panicked at that time because we that kind of tracks for monkeypox. So the thing that made people worried was that a completely separate household cluster was seen. So the group of people who were who on the 12th of May were detected with laboratory confirmed monkeypox cases, and they did some
Mm.
contact tracing and didn't find anybody who had been traveling to an endemic country. There's no source of infection that can be identified through any of their contacts as well. So
Yeah.
so of the 98 people who were in contact with this household cluster, they couldn't find anyone else who had monkeypox. They couldn't figure out where they'd actually got it from,
Yeah. Yeah.
which
It's like
is
it's concerning when the reason isn't 100% known because it might mean that there's background stuff happening, right, in an unobserved population that is then seeding these infections. So that's the concern point, I believe.
yeah, exactly, because they didn't get it from
Hmm.
nowhere, but because we don't know where
Yeah.
it is, we have to assume that it's somewhere out there in the community.
Yeah.
And further cases were confirmed on the 16th of May, where there are more of them reported amongst adult men. And this is when things escalated because now the health systems were alerted anybody who shows symptoms of monkeypox. So monkeypox, they tend to be like small pustules that they can be
Hmm.
mistaken for other diseases. And usually people wouldn't test for monkeypox in the UK because it isn't endemic here. But now that
Hmm.
people were alerted to that idea, they started testing more people, and people were coming to sexual health clinics, people going to turn up in hospital. And as of the 25th of May, 86 cases were laboratory confirmed to have monkeypoxes. And
Mm.
around 91% of those cases were based in London, which is the
Yep.
city where I live. So I'm going to be a bit more careful. Um, of
Yeah,
these
actually,
cases,
I just want to say that that's like just that story in of itself is like a wonderful tie into like what we always close our episodes with, right? Like asking the right questions is so much about science, and like this part of like infectious disease surveillance is helping, right? Clinical scientists ask those questions in the right way, get those test results to understand what's going on, and give us some context.
yeah, exactly. And London is a big city, so there's lots of
Hmm.
opportunities for infectious transfer.
Hmm.
So they looked at a cluster of people, and they found that was it among the cases where was information was available, they're all men, and I
Hmm.
think 66 cases were classified as GBMSM. So that's men sleeping with men, so that covers gay and
Yeah.
bisexual. Um, so that and this is a community who is tends to be quite kind of I guess clued into the worries about sexual health and infection. So if anyone's going to go to a sexual health clinic, it's usually that community because
Hmm.
of the big scare over the HIV outbreaks. And
Yeah.
I think people are very aware of those disorders, somewhat more than maybe other communities.
Yeah, and I think it's also it's like it's maybe also indicative of like a robust sexual health like understanding, right? That like regular testing and like making sure that you know like statuses of your partners and things like that. Um, yeah, that's what we've that's the community that's gotten, and yeah, thanks for sharing. It's nice that there's definitely a lot of discussion I think going on about this particular outbreak, and this is one report that we have, yeah, that we're showing right now. Fuzz is talking about a report about the UK, but of course, this is just one of the outbreaks, I suppose, that's being tracked.
Yeah, and another thing that so there was like a set of responses that UK did in response to this, or feed into that other outbreaks.
Mm.
The first thing was to try and just define what kind of health response they'd do. So one of the things they implemented was like say ring vaccinations. The idea because monkeypox is closely related to smallpox, and so the actual smallpox vaccine does work against monkeypox. So they offered people who were in contact with people who turned up with infections vaccinations. This is I do want to highlight one aspect of the paper that is kind of interesting to me is that the uptake of these vaccinations where it seems to be quite low. Um,
Mm.
so around so with healthcare workers, about 69% of healthcare workers who are exposed decide to take the vaccine. About 14% of people who are exposed decide to take up the vaccine. Um,
Mm.
but even with that, they were still put in isolation as well, and
Mm-hmm.
and I think that so the question. So we'll have to see how well this kind of control is working because the time frame that which monkeypox works on is about in the frame of like months. So
Mm.
people who maybe were infected by people in say May might only be showing symptoms like in kind of mid-June perhaps or later. So this is going to be kind of a
Gotcha.
slow burner in terms of seeing how long it takes for this infection to build up.
Mm-hmm.
Um, and it can be quite difficult to identify contacts sometimes because it's a big city. You brush past a lot of people, um, and I think
Yeah,
that brings us,
totally.
yeah,
The
that
next,
brings,
so the next paper is another surveillance report, right? Like this time based in Lisbon.
yes. So like this outbreak has not just been confined to the UK. On the 3rd of May, there are like 5 males with uh, strange lesions that they didn't know what. So the university was looking into that, and they didn't know what they were. They assumed they were a sexually transmitted infections of some sort. So these people turned up in like various like sexual health clinics as well.
Mm-hmm.
Um, and the thing that clued them into sexual health was the fact that lesions predominantly appeared around the genital area. Um, so they were kind of baffled by these cases, like what was happening. And then they heard about the cases in the UK, and the UK sent out an international alert to be aware of monkeypox. And so they tested these patients for the monkeypox and found that they were positive.
Mm-hmm.
And since then, they've again send out that massive net to monkeypox, and they found that there are like lots of people in Portugal. So like UK currently has 225 monkeypox cases. I think Portugal currently has around 100 and 143 confirmed infections so far. Um,
Mm-hmm.
the interesting thing
And
here is
what
they also tried. Sorry.
Oh, I was just gonna say, I feel like, and once again, like what makes that something of concern for public health officials is not having like the full understanding of how the transmission is happening, and so it gets categorized as community transfer.
Yeah, and this paper tries to go into try. They tried to do a lot more in trying to find out like what the exposure routes are. So
Hmm.
um, they looked at saunas used for sexual encounters. There's travel abroad, Spain, UK, and Brazil. What other things are there? And they tried to screen through all these contacts, and they found like one case was a contact with another confirmed case.
Mm.
So there is this missing transmission that seems to be happening that we're not quite able to understand.
Hmm.
Um,
And in some ways, it's not ever going to be 100%, right? I don't think we would want that necessarily out of our healthcare systems, like, or at least like there's like that's like a privacy trade-off, right? Like how much personal information do you know about people's lives? I'm thinking back to like when we were looking at the SARS-CoV-2 outbreaks and people were using like cell phone movement data to try to understand like where those transmission rates are happening. Like, yeah, you can get better, higher resolution views into the population, but then like sometimes those data sources are not what people want to be tapped. And so I guess what's really nice, what I think is a positive about sort of sending up the flag right now in terms of this monkeypox outbreaks is it gives, hopefully people are clued into the news and like listening to what's going on. It gives people something to look for, right? Like this is like, oh, if you see these types of lesions, if you have something and like your doctor doesn't know what it is, like here's something now you can like maybe like tap into because it could be something that's going on in the background and there's not enough awareness yet.
yeah, and yeah, just for the final thing, I'm going to bring up there are cases in the US. So
Hmm.
monkeypox outbreak in 9 states in the United States. Um, so again, the US did have an outbreak of monkeypox back in 2003, which was apparently carried in by infected prairie dogs.
Mm.
Um,
So not just it's definitely not. I think the monkeypox is a funny like naming convention, right, for this particular thing. Just
Right.
like first found in monkeys, but actually, I think what I was reading was it was saying they're not sure what the reservoir, the zoonotic reservoir, is exactly for monkeypox. But yeah,
Yeah.
but it is a zoonosis in that it spreads from animals into humans. But then there is, and clearly because of the outbreak information that we've seen, there is human to human transmission on the back of that zoonotic transfer.
Yeah. Yeah, I think we'll go into that. I mean, here like we have people who were traveling around the world and then came back with rashes that were later found to be
Hmm.
monkeypox.
Hmm.
So again, kind of a uh, similar kind of pattern that we've been
Yeah,
seeing.
and we have a
Um,
picture of some lesions in this particular paper.
yes, we do.
Uh-huh.
Uh, okay, if any of you are eating right now, then maybe you'd want to kind
They're
of
not bad.
pause the
I
video,
don't think they're that
finish,
bad,
and
but yeah, we also have different. Everyone has a different
yeah,
attuned sensitivity for these things.
yeah. So these are kind of what the lesions look like. So some of them look like kind of they can be mistaken for severe looking. Um,
Hmm.
but yeah,
Yeah, they
that's
kind of like wart looking,
yeah,
right? um, and then it sounds like based on the description, and so the course of these things are to sort of get scabbed over, and eventually like the scab falls off, and you just have like a dimmer and dimmer scar of where that pox pustule was.
yeah, exactly. So yeah, the question that kind of comes to mind is why is this suddenly happening now? Why is
Yeah.
and the thing is like it might not just be a sudden thing that's happening now. So I'm going to dip back for a paper that was published all the way back in February, um, called "The Changing Epidemiology of Human Monkeypox: A Potential Threat," which is a very prescient title for something that was published in February,
Totally.
not through. Yeah, um,
But
yeah.
uh,
They
but
the
they,
people in this paper.
yeah, but this is one of these in-depth literature reviews where they dig in to find all the papers that have been published associated with monkeypox, as far back as I don't know how far back they go.
I think
2010? No.
yeah, they go back pretty far. So they look at like picket cases in like between 2020. So they look at it from like 1917, I think. Um,
Oh yeah, so they're going back as far as they can in the literature.
yeah. So I mean, we got this. So essentially, what happens with monkeypox is since it's a rare disease, when it turns up, people often like write a nice case report about, oh, what a rare disease, what a rare infection, and then suddenly you can notice a trend of these case reports becoming more and more frequent. And so this paper kind of pulls everything together to try and put build a story out of all these all individual articles.
Hmm. Hmm.
And
Oh yeah,
yeah,
they I was gonna say it's like these little like yeah, these images, these are those reported cases, and they divide them out by decade essentially. Um, and so yeah, so between figure 2 and figure 3, that's like the 70s to the 80s if you flip back between those two.
yeah, yeah. So like figure two, you've got a picture of like between '70s and '80s. It first really appears and gets foothold in the Democratic Republic of Congo. It doesn't really
Mhm.
like stay very well in other African countries. So when we say like, oh, it's endemic in Africa, I think there is this kind of thought that, oh, it's everywhere, but no, it's actually just in maybe one country at a time.
Mhm.
And then if we kind of fast forward 19, it's in 1980 to 1989, you can still see like, oh, it's still got that foothold in one country in Africa, but hasn't really gone out into the other areas of Africa. Um,
Mhm.
and yeah, you can even see that it's mostly in the DRC. Um, and
But I
that's
but
kind
but
of
I do
sees
but I would like to say that like there's a sampling bias here, right? That this is like it's these are published papers, so
right.
like it has to be something where there's a robust publishing community that's and then
Mhm.
also a surveillance community able to confirm the cases and then publish about them. Um, so it could be more widespread, but like this is what this is what the view we have from the literature.
Yeah, exactly right. Start to see this massive jump in cases of monkeypox between 2010 and 2019. Uh,
Hmm.
so like in there, so suddenly the from like the initial place in DRC suddenly it goes to Nigeria, and there seems to be a much bigger foothold in Nigeria. And this is almost all like accounted for by this outbreak that happened in 2017 uh,
Hmm.
towards the like kind of end of the 2010s.
Mm-hmm.
And they found like kind of the clade that kind of emerged in West Africa is slightly different from the one that is endemic to Central African Republic.
Mm-hmm.
And like it tends to be like the people to get infected tend to be male. They tend to and there are some other interesting tidbits here, like that. Well, let me see if I can pull up the data, but about like 62% of cases have in the Nigerian outbreak have no known transmission network,
Hmm.
which is fascinating because that means that there is something about the way this version of monkeypox is transmitted that we are not particularly sure of.
Mm-hmm.
Although I think there is an elephant in the room that I will talk about in a little bit with regards to this.
Mm.
Um,
What's
but I think this kind of
so in Figure 5, there's like 47 cases from the United States. Is that related to a known outbreak of some sort? Like I don't.
that was the 2003 outbreak,
I see.
I believe that we talked that was related to the prairie dog.
Gotcha.
Um, so that was like a zoonotic outbreak. Um, so yeah, that was a pretty big one.
Right. I see it. Then it just doesn't happen. So yeah. So it doesn't. Yeah. It is. It is. Hmm. But prairie dogs are not native to Africa, are they?
Yeah, I need to look up the details of that case
Yeah.
because
Yeah.
I
That's
think that
interesting.
essentially what happened was I think there was got caught it off another animal that was imported from Africa, and there was like this long transmission.
I see. Okay.
So
Cool.
yeah, uh, and then we go to like another paper that was originally published in 2020, but it was actually put in issue in April 2022, uh, called "The Exportation of Monkeypox Virus from the African Continent." So this one describes like some of the cases that were like kind of exported from Africa early on, like during. So there are a couple of cases that were exported to the UK and various other countries during 2018.
Hmm.
But between 2018 and 2019, obviously, like 2020 to 2021, there's a massive decrease in international travel. So maybe that. But I think the suggestion here is that like again that we have like lots of travelers going out, and that the kind of they're trying to link the kind of peak in the export of this virus to peaks in the infection in Nigeria.
Hmm.
So there was this massive like so let me see if they've got a graph of this, but they
Yeah, the thought is again, it's like it's trying to build this like where does it come from story so that like people can understand like where they can then control, I suppose, that spread. So the idea would be that if you see like an increase in cases from Nigeria and then those genotypes are closely related to the cases that you see in other parts of the world, then like you can build that association.
yeah, so you've got like so they're mostly looking at like exportation related cases, but the US outbreak was kind of very different kind of set of cases, and they're
Hmm.
trying to build up this story of like what how can we stop this from getting out of Africa.
Yeah.
um and
Yeah, so here I guess like the reason why they have the exportation related cases is because the couple of cases that genotype here in the UK in blue are highly related to this one that's in Nigeria in red.
yeah,
Yeah.
that's right. um yeah, and I think the thing that yeah so the thing is like this outbreak isn't necessarily unexpected because there were like kind of instances where this could have spread much earlier, and I'm
Hmm.
getting the sense that the only reason this hadn't spread so far is because we had all those travel restrictions in 2020 and 2021, and this is something that was kind of
Yeah.
always in the offing.
Hmm.
But another thing,
Oh, I just want to say in this paper, the USA strains are used as
yeah,
references, right? So they're specifically looking for the link between UK cases and Nigerian cases in this
yeah,
study, because like they're saying like because I guess the U.S. outbreak, it's like known the source that it came from, that it's known like to be genetically all related. Then like they're using that to interrogate some of the other branches, pushing them together.
yeah. and like those US strains are probably some of the few monkeypox strains that were like sequenced to a lot of depth
Sure.
and kind of were
Hmm.
recorded. Um, but yeah, I was looking at this paper because kind of trying to figure out like if we want to figure out the future of this outbreak, we want to kind of keep an eye on how Nigeria had to cope with it.
Mm. Hmm.
Um, and I mean, this year the recorded cases in Nigeria are 247, and for reference, so far the UK confirmed cases are 225. So we're currently like, so it's very early on in the outbreak.
Hmm.
And I think another thing I want to highlight is that the reason why there might be a lot of missing kind of contacts in the Nigerian outbreaks is because I'm bringing in the social cultural thing
Yeah.
here, but in Nigeria, homosexuality is
Mhm.
illegal, which means that if it is spread through that kind of like contact, then in that kind of area where
Well,
there's a lot of
not just that
homophobia,
type of
then
contact, but like just sexual contact in general.
yeah,
Yeah.
exactly. Then people might not be coming forward about
Hmm.
it, and so that means that there is a lot of missing information, uh, because yeah, homophobia can kill. I mean, we've seen this from the AIDS outbreak.
Yeah. Yeah. Yeah.
Um,
I'm interested to. So it sounds. I guess what we don't see here is like from the current outbreak in the UK and Portugal or whatever, like seeing this type of study that you picked out done there, right? To get a sense of like what's related to what in the current outbreak.
it's funny you should say that because so there's this place called NextStrain which has been cataloging some of the monkeypox virus genomes that have been
Hmm.
released, and so we've got this little like link to their website, which gives us a genetic epidemiology of the monkeypox virus, showing the the relatedness of the viruses that have been found in the current outbreak. So we've got most of the genomes seem to be coming from Portugal,
Hmm.
so you can see that they're all linked right out here, along with ones from say the UK a little bit, but also like ones from the USA.
Hmm.
Um, so what we're finding is that I think like there's a stats news article that kind of indicated that maybe there might be more than one kind of variant out
Right.
there for monkeypox
Hmm.
because there are like these extra branches that are quite far from the
Yeah.
tree. Um, at this stage, I would be kind of cautious about that because with monkeypox genomes are a lot more complicated to sequence than say
They're
SARS-CoV-2,
big.
so
They're
there
bigger.
may be a lot more like kind of stuff that needs a lot more screening that needs to be done, and a lot more kind of quality
Hmm.
control just to make sure that these relations are exactly how we're
Hmm.
seeing them. But still, that does look like kind of, and we've got
Yeah.
like a kind of a map showing the spread of these different variants. But
Yeah.
um,
So, I mean, one way of thinking about this is it could mean that there is already like there's low-level endemic monkeypox spread in the background of a bunch of countries, and we just haven't been observing, right? We haven't been testing enough for it and understanding like that is like a ongoing community thing. But of course, like so that's just one explanation, right? The other explanation is there is an ongoing, right, like chain of transmission that just isn't being that we don't have all the information on yet, right? And it does start in Africa and it ends in these other countries.
yeah, and another thing that this graph kind of shows is that there's the rate of mutation for this strain monkeypox seems to be a lot higher because like the strains that they've been finding are a lot more genetically distant from the strains you'd find in Nigeria circulating around this time.
Hmm.
Um, which is strange because because DNA viruses tend to have very stable genomes. So like things like smallpox, they don't mutate very quickly.
Hmm.
Um,
Well,
so
I mean, to me that also ties into like that one of the ways that you could interpret this is that like there has been community spread of monkeypox for some time and it just hasn't really been addressed.
yeah,
Yeah.
which brings me to the next not so much paper. This is, I guess, more like a kind of a blog post by a couple of people who were working with the sequencing data and they've got like kind of a hunch about what's going on and why there might be this extra kind of
Mm.
mutations. Um, so what they've noticed is that there are certain mutations that appear more often. So this is titled "Initial Observations about Putative APEC3 Deaminase Editing Driving Short-Term Evolution of monkeypox since 2017. Um, so what they've kind of noticed that there are certain types of base. The idea is like they see like for TC turns to TT or GA turns to AA. So uh, these kind of
This
mistakes are
I was gonna say this is the reason why it's such a specific specific changes that happen is because this APOBEC thing it encodes for an enzyme, and that enzyme does these changes, and it's thought to be an antiviral enzyme. So the idea is that, oh, if you mess up certain genetic things, then like the viruses will just like fail at some level. But like evolution is a long process, and so the history actually seems like there's a coevolution, right, between these enzymes and viruses, in that like some viruses use it to sort of gain some diversity, or like they can use that, right, as a method to gain some diversity. But it's thought to be a protective mechanism, sort of
yeah
messing up random viruses.
yeah, APOBEC3 is kind of like because I mean it's a target that we use. And remdesivir targets the virus genomes and to make it like not work properly. I think and APOBEC3 tries to do the same thing by inducing like kind of a higher mutational load for a virus, so that when it has
Yes,
enough
yes.
mutations, the virus stops working correctly.
Hmm.
And
Hmm.
APOBEC3 works quite doesn't work great against all viruses equally. So for some like viruses like say like some enters into the capsid and is like traveled around by the virus, and so when the virus eventually tries to
Hmm.
infect another cell, it's the FX3 is still there to like to fight the virus.
Yeah,
Um,
I think that's in the that is an HIV story, right? That's from
yeah,
HIV.
I
Hmm.
believe so. Yes. Whereas like things like variola virus, they're DNA viruses and they're more like stable.
Yep.
Um, and so they have lots of different copies of like, and so it is possible for APIC3 to act on lots of them, but then they can still have a stable genome, but incorporate some of
Right.
the mutations caused by APIC3.
Hmm. Hmm.
So like in this case, what they're proposing is that the evidence they looking at the patterns of the mutations in the genomes, they think that these viruses have survived subsequent attacks by APIC3, and that is why they've got all these strange mutations. And part and it's And so that's an interesting explanation. They kind of theorize that this is because it has human to human transmission, and that maybe the virus
Hmm.
isn't used to the human APEC3, whereas maybe it would be more used to APEC3s in its original animal host. And that
Whatever that
this
may
might
be. That unknown,
might
that
have been,
unknown. Yeah.
yeah. And the idea is that this is might be like kind of the catalyst that some other changes happened in the virus that made in 2017 to make it better at
Mm.
infecting human hosts, and that the APEC3 is kind of evidence that something has happened and that it's been spreading from human to human.
Oh gosh. I hope not though. I don't want another virus to have to keep track of.
Yeah, I mean, we've already have like multiple like coronavirus variants to deal with, and now we've got monkeypox. And but thing is, monkeypox shouldn't be evolving this fast, so
Mm.
there must be some reason for it.
Yeah. And again, that reason could be that we have vision on it now, right? Like this is a thing where it's like, oh, now we know it's been happening in the background, and actually vision in this sense is going to be a good thing, right? Because it means that people know what to check for, right? And ask questions when you go into the doctor. The doctors
Yeah,
themselves know how to deal with things. Yeah.
I mean, like this is like all assuming that they are rooted in the same ancestor that was from discovered in Nigeria or from other places, but it
Hmm.
could easily have been spreading directly from the animal host or through. It
Yes.
could be like we've only got the evolution history based on the most recent strains we found, but like it could easily have been like
Yeah.
been another zoonotic event that
Yeah.
has led to this, and that
Hmm.
it's actually there's a lot of stuff that we don't necessarily know
Right.
about what's been happening in monkeypox, and a part of that is because researchers haven't been looking at. We've had quite a long time to get a handle. Been that kind of uh, push for it, and hopefully we're going to get
Hmm.
that now.
Yes. Yes.
And I'm hoping
Yes.
that like this might be the one moment where we decide, oh yeah, maybe we should look out for like neglected tropical diseases that might end up spreading to everywhere else because
Yeah.
Yeah, I mean,
Well,
I'm happy.
we've said it before too
Yeah,
in the sort of vaccine equity thought, right? In terms of
yeah,
like more vaccines for SARS-CoV-2 around the world is like better than boosting people in like localities because like lowering the global burden is lowering the space in which viruses can play around, and it's also just like it's like I feel like it's like very morally motivated, right? Like we want to make sure that we're keeping the world safe because we're all part of it. So
yeah. If we want to stop global pandemics, we need to think about health on a global scale.
yeah.
Like
Hmm.
being
Hmm.
being ready to combat a virus that could kill, like, say, 6 million people around the world. Maybe like going to places where that's likely and stopping it before that happens would be
Yeah.
really worth the money.
Yeah, yeah, yeah. And yeah, and I would say even outside of that, like it's to me that rings true as a very nice way to approach global health, right? Where we're caring for other people even if they aren't in our immediate area because we're all just part of a single ecosystem. And viruses, I think, are just a great way of maybe showing that because there's so many viruses that impact, right, many different organisms around the world, and that gives us a very tangible, in some ways, tangible biological link to different life forms. Because we talk about zoonotic, like those aren't even humans that we're thinking about now, right? Like we're thinking about like just like the health of our of the animals that we live in close contact with.
Yeah, I mean, my perspective is we're willing to just spend trillions to like stop the next 9/11. Maybe we should spend at least 1 billion to prevent the next COVID-19.
Yeah,
It's just my thoughts.
yeah. Preventative medicine goes a long way. Okay, and that's the end of our monkeypox, right?
Yep, that's the end of the monkeypox. And speaking of COVID-19,
Yeah,
um,
in terms of other global global health issues that we're grappling with, I tried to catch myself up on what's going on with SARS-CoV-2 because I got SARS-CoV-2 like 2 weeks ago. Very frustrating moment in my life. Granted, I was not being as careful maybe as I could have been. We also, I mean, I think many people have this feeling now around the world that some of our Swiss cheese barriers have been removed. We're heavily relying now on the Swiss cheese barrier of vaccines and sort of self-isolation techniques. So anyways, I got it a couple weeks ago and I was like, okay, well, what's new? What's new in this world? What virus did I most likely get? And so the first paper that we have for you guys is actually just the published version of something we covered in February. So Virological, the SARS-CoV-2 Omicron BA.2 spike. And so we covered this in February as a preprint. And at that point, BA.1, the original Omicron variant, was sweeping through the world. But on the horizon, there were reports out of South Africa that there's this BA.2 variant, a subvariant of Omicron, that was gaining some traction. And yeah, and I guess like I mean, I guess people, we're kind of behind here in the news, in the story, in the story of the news. Um, but so BA.2 was going up in Africa, and if folks remember back in February, we were saying though, but the rates were going down in general in Africa. So right, like, and so the idea was that while this genotype was taking over, it didn't seem to be causing like a spike in any way in the cases of South Africa, but it had yet to come. It had yet to come to the United States, and I think yet to come to the UK at that point as well. And yeah, so we've already seen this paper, right? That BA.2, it seemed to be more. They go into this idea of like, does it create the fusogenic? Is it causing cytopathic effects, having like the cells fused together? I think if you scroll down to Figure 3.
Figure 3, yes.
Yeah. Figure 3, like these like green these green images, they're looking at whether or not like the cells are fusing together into these syncytia. That's like, again, talking about a characteristic of the spike protein, right? Its ability to fuse membranes together. But like that's just one element of spike. everyone's obsessed with, we know a lot about spikes, right? So it's been like split up into a lot of different subcategories, I guess we can call, of phenotypes that it could possess. So one of them is this fusogenic one, but then the other one is like processing by proteolytic enzymes, right? That's like the cleavage site, its ability to be more infective with TMPRSS2 or other proteolytic enzymes, its ability to just cause a lot of different plaques, right? So there's many different ways to evaluate it. And what folks were saying was that BA.2 was going to be more infectious than BA.1 in this context. It was, I think, where BA.1 was less fusogenic than Delta, BA.2 was more comparable on that front. And I mean, now we're talking about the past, right? Like this was done in February. These were observations being like, brace for it, right? Something's happening. We were seeing, though, the rates go down in South Africa. But actually, at the end of the day, at least from what I've seen now, the current rates in the States, BA.2 is the dominant, and there's even a subvariant of that, BA.2.12, that is even better. And so that's what we're experiencing right now. That's probably what I got.
Yeah.
I got.
Yeah, I mean, I think that this paper kind of like really like delves in, and you see that it confirms that BA.2 is much more kind of infectious than BA.1, and they do a lot of other kind of things. I think they do experiments like the neutralization assays, and I think also like just trying to characterize it. And
Yep.
so
Yeah, and they put it inside of hamsters, right? They showed that it like does hamsters. It's, I guess what's very nice, I guess, to see now, like when we read stuff about new variants, is that there's a suite of assays that exist and methods to go in and like characterize the new variants really rapidly. Like people reach for the Syrian hamster model, right? And say, okay, let's look at this. People reach for cytopathic effect. People reach for plaques, right? Like all of these assays now are like very strongly in the wheelhouse of infectious disease researchers for SARS-CoV-2. And with something new emerging, like we have the tools to characterize it and say, okay, here we go. Of course, we don't know what that interaction will be on the human population per se, but like it gives us a lot of it gives us confidence to say like, yeah, we should expect something to happen, right? We see all the cross reactivity, this is like pathology specifically of the lungs.
yeah, so you can see like, oh, there seems to be more of this than this. So
Yep. Yep.
So yeah.
Okay, so again, that's like that was published recently, but that was news from February. So catching us up to this BA.2.12, and also two new variants now, again being surveilled in South Africa, BA.4 and BA.5. And so so if we go here to figure 1, we can see the whole family tree, the updated family tree of what we're looking at, right? It's all this Omicron stuff. I guess it's a very fruitful branch, and because like these strains are sort of circulating with impunity, I would say, around the world, they're being allowed to explore all the different spaces that they can make a difference. And so maybe so BA.2.12 is becoming quickly the strain that I think is most popular, at least in the States it is. Here, yeah, you can see that here now in D. They have, oh no, in D that's the relative, that's the spreading rate. That's like also like kind of touching on the epidemiology. They don't have the numbers here.
Yeah, BA.12.2.1. That so
Yeah.
like.
Oh, here in
yeah,
E.
I mean,
E has. I think these are the actual numbers of infections.
yeah.
Yeah.
So they're kind of characterizing how you see like BA.2 it had this nice peak, and now you've got like the variants that are derived from BA.2 taking over. So in South
Yep.
Africa you can see BA.4 and BA.5 kind of coming up, and then in the USA you can see BA.2.12.1.
Yeah. Yep. So yeah, I guess, and we've touched on this before. We touched on this. I thought this was actually a really great paper that we should probably fetch it out again. But in all these infectious moments in a country's sort of epidemiological history, like there's an opportunity to do a pairwise comparison between the strain that's dominant and the strain that's coming in. And in that paper, this was some time ago we looked at it, in that paper they showed that there's no clear relationship between whether the up and coming strain is going to have like less severe characteristics than the strain that it's replacing. And I think that like that's something that we're seeing in action now. Sort of like that was something that observation was looking at the past and looking at like, you know, BA, like alpha to beta and like delta to Omicron and all those different ones. Now we get to see it within the Omicron lineage itself. The same relationships are sort of being set up, especially in the sort of predictions that are being made in terms of the, I guess, severity of BA.2.12 and BA.4 and BA.5. I'm hoping that vaccination rates are going to be like a big factor that suppresses, right, like the growth of these and the spread of these, but like I don't think they can be, you know, solely relied on. Like, it's still good to exercise some caution. BA.4 and BA.5, I want to point out. Oh, sorry, do you want to say something, Faz?
Oh no, I was gonna say like that the vaccinations do seem to still have some protective effect compared to like say like
Oh yeah,
unvaccinated.
definitely.
But I don't know if they said that in this paper or not. So I was being precautionary,
Gotcha.
and that's why I kind of
Gotcha.
paused.
Yeah,
Um,
I think that like none of these papers are looking at the vaccine's effects on the severity of these things, right? These are all just like, as I said, there's a whole suite of tools that scientists have now to do predictions, right? And just say like, oh, okay, based on how well does it fuse with cells, based on how well does it evade different serum, right? Like that's sort of these papers deal with that. They don't actually look at like like, because you only see that in post hoc. And I mean, we probably have that now for BA.2. I didn't pick those papers out though to cover. I guess the other interesting thing I want to point out here in C in that figure 1 still is that with BA.4 and BA.5, so these are new ones that are being surveilled right now in South Africa, the S genes come back. So like, I think we talked about this a little bit, that like when Omicron came on board, it was like, oh, like the S gene's back, and so like this can be used to surveil it. And then when the BA.2 came, it's like, oh, the S gene's gone again, so you could surveil it that way. And now BA.4 and BA.5, the S gene is back. And so like, again, like I don't know why this one locus is being such an interesting marker, like on and off, sort of oscillating, but it kind of has been very strangely. They also point out like that this L452 mutation, like this locus has been shifting in the Omicron lineage quite a bit. So like it's the same L, the same lysine that's changing, but it's changing to a bunch of different things. So again, I really, to me, when I see this, like I'm seeing like, oh, the virus is really getting to try out a lot of different things in the population to see what sticks. Yeah.
yeah, it kind of also like highlights the importance of that kind of area for the for the Omicron adapting or doing something to.
Yeah. Yeah.
So there must be some selective pressure on that area to
Yes,
change.
absolutely, absolutely. And again, this is like the big mystery that we've been grappling with this whole time, right? Every time a new variant comes out, it has this whole new set of different mutations, right? I mean, not whole new set, but like a whole suite of mutations. And knowing how those all interact together to make the virus better or worse is not something that we can easily solve. So this paper goes in and it does like all the same stuff that we saw with BA.2, right? It's just it's doing that stuff with BA.2.12 and BA.4 and BA.5, and we're seeing that BA BA.2.12 and BA.4 and 5 seems like it's better, like more. It's a more successful virus than BA.2 itself. Um,
Yeah, I think that's
yeah.
the thing. Like, I think it was important to show the previous paper so that you understand that these are found even better than at like kind of fusogenic and kind of causing infections than BA.2.
Mm-hmm.
So
Mm-hmm.
so that we can get
Mm-hmm.
this idea like that there is incremental changes in Omicron that are making it
Yes.
more like able to try and fight the immune system.
Yeah. Yeah, absolutely. Which I think, to me, like says again, like this is the problem with relying on vaccines as the primary like shield against something, is that like the viruses do get a chance to sort of vary themselves. But yeah, okay, that's all.
Yep.
And then these are two more papers from Omicron, but I'll just talk about them very briefly. So this one is this one focuses very much on serum, so differential evasion of Delta and Omicron immunity for Omicron. So again, looking at the BA.4 and BA.2.12, testing it against serum from different individuals. So if we go down to figure 2.
See, figure
Figure 2, yeah.
right.
They have like a bunch of serum from Delta patients, and they're just testing that against the different variants. So you can see, like, we always know this. Anytime they say that things are evasive, like, oh, this is more immune evasive, it's coming from this data. This is the data that underpins that statement in the news, right? Is that like, yeah, serum just doesn't work as well with some of these. The cross protective is variable, right? Like, there are nuances here that I guess I could talk about, but I'm not interested actually. Like we know the rigmarole of it.
like strange things like, say, like the BA.2, it quite evades the Delta serum, but then like BA.4, 5 and BA.12.1, they actually tend to be more
Yeah,
neutralized
more
by it.
neutralized by
So
the Delta serum. So it's like, okay, so like some things it's better at and some things it's worse, and it's like it's a new mix.
yeah, but I mean, I think that kind of is interesting, because I mean, these viruses might be good in some ways, but they might also be sacrificing something else in order to get kind of
Yeah.
that one up.
Yeah, absolutely. And I guess that sort of highlights the complexity of this evolution. And this is coevolution in practice, really, right? Like it's co-evolving with our vaccinations and our behavior strategy. Like we're seeing this, and I guess after like many years, like there might be like a stable configuration, but we're definitely not in that phase. Like this is still like in evolutionary terms, this is all like the flash in the pan moment, right? That like all the forms get tried out until like yeah, things can find their niche, I guess.
Yeah, I mean, another thing that is like the Delta, the BA.4.5 is very protective against like the people who've been infected with the BA.1 variant.
Mm-hmm.
So that kind of speaks to maybe that's why that's kind of emerging in South Africa, because South Africa had the BA.1 variant mostly, and in order to survive, any new variant has to be able to counteract that prior immunity.
Mm-hmm. 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 Mm-hmm.
So that's just like an interesting niche, because I think like the way that the virus has to transmit now, it's in such a different context, because now there are people who have kind of this previous immunity from either vaccines or previous
Or both. Yeah,
infections.
vaccines or
Yeah,
yeah.
or both.
Mm-hmm. Mm-hmm. And it's good. It's good that I think that we're throwing up booster shots and things like that, right, as different barriers. But there is that like very reactive. It's a very reactive way of dealing with this, right? Where like de novo, we could have said like, let's just make sure that we pursue, like we just stamp down the virus across the world. But yeah, it's just like logistically that didn't work out.
Right.
um,
Yeah, and I think we're going to like take that into account with whatever we do next time.
Yes, yes,
Um,
yes. And the last paper that I'll pull out, and I didn't really want to talk too much about this one, like just like I'm kind of just like blowing through different Omicron papers, but this one, delineating antibody escape from Omicron sublineages. In this one, they're actually they look at serum again, but they're also looking at monoclonals in this one, right? Like there was that like a whole panel of monoclonals that people can look at to investigate whether or not a variant is becoming more immune evasive. But what I really liked is in figure 1, they have a nice like BA.1, BA.2, BA.4, and 5 all of the different mutations, also the original Wuhan. And I just like this table a lot. No, the table above. I mean, you can see it in those ones too, but the table is nice, easy to read for me. And like, yeah, so this is like a bigger version of that first one I fixated on, which had like a smaller table. You can see like all these different mutations, how the Omicron strain is so different from the original Wuhan object. And even now within the Omicron, like there's actually quite a few differences. Like you can see there's a stark difference between BA.1 and BA.2 and 4 and 5. So again, I just wanted to sort of impress on people that sense of like how the virus is finding its spot and needing to find its spot through iteration. And then what I also thought was interesting, oh no, actually, maybe I misread that. I thought the D614G mutation was not present, but actually it is.
Oh, yeah.
Okay. Yeah, it's through all of this. Okay, never mind. I I fixated on some strange detail here that I actually just misread the graph. But yeah, this is this is what I wanted to tell people, like just to look at like the way in which things have changed over our 2-year experience, 3-year experience, going on 3-year experience probably with this virus.
Yeah, and it's quite neat. They've got these graphs that show like the changeover between variants and how that might be affecting things. I mean, and you can see like the outbreak peaks, and you can see like how things changed in like UK and various other countries in terms of what. So like how BA.2 and the Omicron has taken over, and now you're only just seeing like in other countries that maybe we're starting to see BA.4 and BA.5. It's and BA.2.12.1 seems to be like
the fact that the United States has its own variant now, right? Homegrown Omicron variant. I mean, again, I think that speaks to me. That speaks to, and again, I'm not. This is a horribly unscientific thing to say, but like, I just like, to me it speaks to the fact that like there's spaces for these niches to develop, and it speaks like like not good control, not good control in my opinion.
I mean, yeah, I was here when the UK variant appeared. So I mean, the thing is, you can't control where a new variant appears, but yeah, no, I mean, I'm not saying that the UK response is much better than the US response, but I do feel like that it is unfair to say that, oh, a new variant has appeared in a country. That must be the country's fault.
Right.
So,
yeah. Okay. Yeah. That's true.
I'd say that for every other country, so I'd still have to say that for US.
Yeah, that makes sense. Yeah, I just wanted to, like Minref was saying, like, yeah, no Delta. Delta is definitely a thing of the past, but that idea that Delta was more fusogenic, right? And that was like a really scary thing. Like, it's interesting to see that in the Omicron clade, like that's a convergent thing, right? Like, it's not that things have picked up. I mean, there are some reports of these of recombination events, but actually, from what I've seen, it seems like it's a convergent thing. That like it's another, like, fusogenic is just another variable that the virus can turn on and off, and depending on whether or not that serves the virus in terms of its spread, like that's what we'll see in terms of the ability of a spike to adopt that strategy. And it does seem like going back to fusogenic is something that the virus is not done exploring.
Yeah, but I mean, it's not all like doom and gloom for this paper. They do like screen out a bunch of different like monoclonal antibodies because to figure out which ones will work for the new strains as well.
Yes. Yeah. So I mean, I guess the antibody, like the reason why people put the monoclonal panel, put every new variant through the monoclonal panel is to evolve the clinical side, because although now with the proliferation of some antiretrovirals, that's also the good news sort of layers, right? Is that like there's also antiretrovirals that should be insensitive to the changes in the spike.
Yeah. I mean, a lot of them target the RNA polymerase, so like things like remdesivir should still work the same way. Molnupiravir still should work in the same way. So I think that's there's still like stuff that works.
Yes, there's still stuff that works. I guess, yeah, I definitely don't want to be too doom and gloomy about it, because like there are so many tools to reduce deaths. Although maybe those tools aren't like aren't equally distributed amongst aren't equally distributed, but like they do exist.
Yeah.
Okay. Okay, so that's the end of SARS-CoV-2. And just before we leave, we did want to still cover 2 more papers.
Oh,
You feel
we
like we
have
have the time
one
for that,
more
Buzz?
paper on SARS-CoV-2, daily longitude.
Oh,
That was for the
right.
new outbreak, or the like kind of new stuff about like what's going on with Omicron. But now we've got like some things about like normal SARS-CoV-2 infections. Like, well,
Yes.
a paper on the daily longitudinal sampling of SARS-CoV-2 infection reveals substantial heterogeneity in infectiousness.
Mm-hmm.
So,
Yeah, actually, this one I was reading when I was infected, just because like I wanted to know like I wanted to know my testing regime, like whether or not my testing regime was fitting in with like what others were seeing, because like in this paper, so these are all non-vaccinated. This is actually a very in-depth data set, which I thought was interesting. So they're following people at the University of Urbana-Champaign. This is before the vaccines were really widely available. So everyone who was enrolled in this, they did not get a vaccine. They also did not go to the hospital. So it's like a subset of people that just did not get bad disease, but they did get disease, and they were also tested. Yeah, that's the figure that I really like, the one that has all of the. It's like everybody had like multiple PCR tests, both from their nose and their saliva, as well as antigen tests, as well as live virus culturing. So the red X's are live virus culturing, the dots are the various PCR tests, and the dark yellow bands, that's antigen positivity.
right.
And so what's really nice about this, and we've said this before, but it's nice to see the data, I guess, is that like antigen tests are really good at showing if you have live cultural virus in you. But PCR tests are great at giving you that early warning to be like, oh, like you're gonna have some live cultural virus. And then what's kind of interesting is there's heterogeneity here, right? And that's actually the main thing that they're pulling out in this paper is you can see some people that don't get much live cultural virus, and then you can see some people that get a lot of it, or at least like more days of it than others. And so like they go and they categorize all these different I guess outcomes that they could have, and they come to this conclusion in the paper that there is also biological variability in terms of like spreading events. That like it's not just like your social network and the differences in the social network that can drive spreading events. It's also like your own response to the virus. Some people are super spreaders. And that's not just because they have parties all the time. That's because like there's something in the biology that drives that.
Yeah. I mean, I've always kind of felt like there is no average infection. Like all infections kind of, there are lots of
Uh-huh.
different opportunities for different paths of infections to go down. And so like if you try
Mm-hmm.
to like pare it down to the average, you're going to like be compressing different like kind of into one that will lose a lot of the kind of important information. Like so you got like lots of people have like say long-term infections here, and some people have like a very short kind of duration
Mm-hmm.
of infection, and grouping those together might not be informative about how everything goes. You need to kind of, so I think that this is kind of a good paper in terms of like the way they decide to model it. So I think they do a lot of
Yep.
molecular model and not molecular, mathematical modeling to
Math,
try and find
yes.
out
Yeah,
what fits.
yeah, like based on the parameters that they picked up in this data set. So like this is their raw data essentially that we're seeing. Their raw data visualized in a really nice way. But based on that data, they plug in some parameters to drive home that point that there's biological variation that also underlies the super
Yeah,
spreading
and
events.
also I think that I should point out that they did find differences in PCR from the nose and PCR from the saliva, in that they
Mm-hmm.
those two areas can come positive for SARS-CoV-2 at different time points, and
Yes, yes.
that is an interesting finding in and of itself. The idea that the saliva and nose are kind of separate compartments that are infected at different times during the course of infection.
Yeah, yeah. So actually, like my mom gets tested quite frequently to visit my grandma in like a care home,
Mm.
and they've checked everything like as their as their screening test. Like they used to just do like the nose swab as people were saying, but then now they like swab noses. They also take some saliva and like the back of your throat. They just want to make sure that like if you're trying to find virus in terms of like and keeping it out of a location, you want to test all those locations because yeah, there's there's no like surefire way of finding it. And I'll say that too is changing based on the variants, right? Like we saw that with the like sort of the different cell in Omicron, right? The way that it was using a different entry method primarily, right? Like that's and that changes like the cell types it goes and changes the location in which sampling happens. I think that that's just
Yeah, and that has ongoing effects on transmissibility.
Right. Right. I guess tying all of this back to like right that mutational landscape. Yeah, nothing here is I think like it is like telling us the story of exactly what's happening now, but it really like fleshes in like the details that you need to be able to make some predictions about what could be going on, and also just telling us like what the most careful way to approach things is, right? If you're concerned about finding viruses or stopping the spread 100%, then like here's the variants that you have to deal with.
Yeah, I mean, another thing, like once they had their model map, they were able to kind of try and pick out some of the other factors that might be affecting infection. So one thing they picked out was age, and kind of based on their models, which were kind of modeling not just the infection but the impact of the innate immune system, other factors. They figured out that perhaps one of the reasons why age is such a factor is that the innate immune system responses might be less effective at limiting SARS-CoV-2 in the nasal compartment in older individuals.
Yeah, so they'd have more shedding, essentially, is what's going on.
Yeah, and it'd be more likely to progress to a full-blown infection because the innate immune system isn't able to control it.
Right, right.
So yeah, a really fascinating paper.
Yeah, I feel like this is like trying to compress this whole month of SARS-CoV-2 papers into like the short little section. Yeah, it's there's a lot of stuff that's going on, and again, like it'll be ongoing. So hopefully, in like coming weeks, we'll just be able to digest this information a little bit better as we see new stuff, especially about this BA.4 and 5. Like I'm interested to see like where that lands and how the characteristics of those spike proteins like end up playing out within like our vaccinated populations and convalescent vaccinated and convalescent populations.
Yeah, and I think like for this month we were trying to rush to bring out the most like interesting stories for this show, but we'll be picking up on some of the other things that have been happening as well.
Yeah.
But I think that brings us finally to the end of our COVID coverage, and we have a few fun papers. So this is what I'd classify as like fun movies from the microbiology world.
Yeah, a series of fun films, short films. But I mean, this is we were talking about this a little bit beforehand, but like science is about seeing, right? Like there's a lot like sometimes we see through statistical inference, but also images themselves are incredibly informative. It shows us like spatial relationships between different entities. And in this case, we're talking about bacteria that live in the mucus that line our guts. So like that location is just, it's full of bacteria. Essentially, it's still part of the outside, right? We have the hole through our bodies, a tube that goes through our bodies. And go ahead, play this. I can talk over it.
All right.
The authors are able to use a technique to make the tissue clear, to clear the tissue, so that they can then shoot their optical microscopes through the tissue and get this like view. And so they stain in blue, we're seeing the stain of the tissue, and in red, we're seeing a stain for the bacteria. And the way they're able to get this big image is only through very careful sample preparation, because there's a lot of 3D space here. And you can imagine having to like use a light microscope to look through something like that. There's going to be I guess cloudy stuff, right? Like we're not transparent objects, but they're able to treat the tissue in such a way that it does use these dyes, these fluorogenic markers to highlight the things that they want to see.
Yeah, I mean, so we've got these like wonderful like videos where they tag different parts. So as I mentioned before, we've got the villi
Mm-hmm.
in the colon, and you got like in the sequence, sometimes they show the mucus as well.
Mm-hmm.
So there's got this another video
Yeah,
which
they're
has
able to fix the mucus, and the mucus is actually it's not a homogeneous environment, right? There's a
mm,
outside and an inside. There's clumps. You can see there's like dense clusters and things, and all of that is impacting the way that bacteria live in that environment. And so the idea is that this tool is going to give us like more insights into the dynamics of this particular environment. Yeah. Yeah, I guess I want to point out here, you can really see there's like a gap between where the bacteria are in red and where the cells are in blue. And that's because like in that zone, there's mucus, but there's also secreted molecules from our cells that are keeping at bay certain bacteria.
yeah. Yeah, it's fun because like usually when we think of the gut, I always something to think of like bacteria like it did straight to the surface rather than like this thick like kind of mucus environment that is around them that bacteria like kind of live in. You've got like mucus feeding bacteria. You've got all sorts of other like kind of things that are
Yeah,
living in there.
it's an aura, right? And the closer you get to the cells, the more you have to be able to evade the specific compounds that are being secreted by those cells. But there are bacteria that live more closely associated with the surface, and I think they have some pictures of that later. I don't know if they have videos, but they definitely have pictures of it later.
Yeah, yeah. You've got because you got like kind of the gaps in between. So gut has like these fingers like called
Hmm.
villi, and you get lots of bacteria that are living in between
Yes,
those villi. We're
the
called
crypts.
crypts. Um,
Figure 5 has a good one. So they also have like you can also differentially tag bacteria. So like different groups of bacteria, right, have different 16S sequences that can be used to attach different dyes. And so in this figure, they have this like mixture of dyes that basically highlight the bacteria in different colors. The different bacteria from different groups are in different colors. And in that J, K, and I think that's a really clear image that shows that there's one clade of bacteria that does really get down into the crypts, the Bacteroidetes. And it's thought because they have a way of interacting in that. Well, actually, the story that I've heard, I'm not sure how much it's updated since I've last like read about it, but the story is actually that the bacteria are able to incite our epithelium to create more like defensive molecules, but because they are the ones that are inciting, they also have the adaptations to live in that environment. So they're creating this niche for themselves, right? By like stimulating our immune system to make more molecules, and they can live in that band. So it gives them their like private private clubhouse in the bottom of the crypts.
so yeah, they're using our immune system as assassins to get rid of their
Yes,
competition.
yes, yes.
So yeah, we're effectively their bouncers to their like kind of VIP room in the crypts.
Yeah, yeah. But again, that story is evolving. But that's like one of the stories that I've heard, yeah, from understanding how these differentials form and what that interplay is with the host.
So, yeah, the red are the bacteroidetes in
Hmm.
this kind of thing. So you can see that like deep down, it's like you got these red dots, and then above you get all the other
Yeah.
multicolored dots.
Oh yeah, they do have a scanning one. Yeah, and yeah, just like
Yeah,
a really cool technology. Hopefully, these authors get a lot of collaborators, and we see these images in more papers to come, telling us the complexity of this particular environment.
yeah. I mean, this is part of like one of my favorite kinds of paper where they essentially have a new technology and they just point at
Uh-huh.
stuff, and then we discover new things.
Uh-huh.
That's yeah.
It's the biology
Yeah,
equivalent of
and
the James Webb.
exactly. Uh, Now we've also got our final paper, which is a coordination of two opposite flagella allows for high-speed swimming and active turning of individual
Hmm.
zoospores.
Yeah, so we're talking about a plant pathogen here, and it's called Phytophthora parasitica, and it's a type of fungal object, but it reproduces through these zoospores that are motile, and so I guess the dominant thought is that their motion is what lets them infect a lot of different plants. They basically swim through the wet spots of the soil in order to make contact with the roots. And so in this paper, they just want to investigate like how do they swim? Because they have like a really weird configuration of flagella where one flagella sticks out of the front and one flagella sticks out of the back. So the anterior and the posterior flagella. And it's that anterior flagella that is covered in these weird structures. They call them mastogonemes, hairs, essentially. They have one front-facing hairy flagella. And the authors this is the EM imaging, so to actually see the structures in detail. But the main bulk of this paper is looking at video of these swimming bacteria using particle tracking and using imaging to look at the details of how the flagella is moving in order to get a model of how they use these two flagella to get places.
So yeah, I think that the focus on isn't just on like, so there's like two types of motion. There's swimming and there's finding a new direction. So like with, I think a lot of flagella, there's like different ways. So like some flagella spin in the opposite direction, which causes them
Yes.
to like not propel to spin around, and then they find a new one, kind of almost
Yeah.
randomly. Whereas this kind of has a different, because that's like two flagella, and I
Yeah,
think
so the classic model in bacteria is the run and tumble method, where like spinning in one direction gives you linear motion, and spinning in the other direction gives you kind of like chaotic like tumbling, rolling around. And so like switching on between running and tumbling at specific moments like can drive objects towards a certain gradient. But I think, yeah, what they observe in this particular model is a little bit more, the turning isn't tumbling as much as it is actually turning. They're able to coordinate the motions of their two flagella. I think they say that one of them stops moving and the other one starts moving in a different way. So you can think of the anterior flagella, I guess, in this particular organism as a rudder, as sort of. Yeah.
yeah, because like the way it looks, because like with flagella, since it's got a rotational motion, there is sometimes like, I mean, this happens with helicopters where you need to have that side like rotate in order to stabilize it. So I almost feel like turning off that one, that the flagella that's trailing, like allows it to kind of use that spinning motion to turn around,
Yes, yes.
and then it engages.
Yeah, that was a great video right there. Like I think that highlights that turning, right? Like these sharp turns that they make, which again, like I guess people didn't expect sometimes at the tiny level. You think it's all like just controlling chaos, but no, they know what they're doing.
Yeah, it looks like there is a lot more intention in this. Like I mean, especially you can see like it's not just the back flagella that changes, but even the front flagella, it starts to like move in a different way. It's not as fast. So like they both kind of slow down to turn, just like any responsible driver would.
Yeah. They're respecting the speed limit of turns. They know they don't want to fly off into the guardrails.
They're not going into turns at like fourth gear. They're going down to at least like second gear maybe.
But yeah, just really great like observations and physics really. There's so much physics. They have like a complicated model that they use to explain like the forces that would be generated by these types of motions and how that relates to, oh, look at those like scientific equations. Indecipherable to me.
Yeah.
But yeah, trust the physicists know what they're doing. And then, of course, in true physics fashion, reconstitute the variables into a model.
Yeah, it's all very important work, I think. Because, I mean, looking at real motion at this small level can yield quite a lot of interesting insights.
Yeah. Yeah, and the physics is different down here as well, right? Surface tension right? Fluids behave differently when you're a much smaller thing in the fluid. And yeah, and again, it's important. The reason why I guess it has some importance into our lives is because this is a plant pathogen. And so this is also understanding the way pathogens move in the environment, and maybe would help us do something in crop health. That's the hope. or maybe tiny robots. There's a lot that happens in bacterial or in microbiological motion that I think also feeds back into like when we want to make our own tiny objects, like here are some lessons, here are some biomimicry targets to look at.
Yeah. I mean, why invent something new when nature evolution is created already? I mean, you can always
Yes.
just steal some great kind of locomotion from other creatures.
Totally. So yeah, that's the end of what we have for you today. And so we want you to join us next week for more cool microbiology news.
Yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yeah, I totally agree. You can reach out to us over Twitter with @microtwjc. We also love that folks have joined us in the comments in the chat to chat. We really appreciate everyone showing up, and we've gone over today, but hopefully it's been interesting for folks. Peer review is a process that we can all participate in, so we hope that you have a really great time hanging out with us. If you think you have something to add or found something unclear, just let us know.
Yeah, it's been a pleasure chatting with you, Danny. It's a pleasure reading all the comments as well. Lots of great material there. Yeah, thank you very much.
Same here.
Yeah, tune in next week for more microbiology content. I mean, oh god, I'm sorry, I'm having some technical issues to queue up our end credits, but now that's fine. Yeah, it's been a pleasure chatting with you. See you next week, so where you can nerd out more about microbiology. Bye.
Bye-bye.